All right. Hello, everyone. This is our fourth genetics video. I will be going through a PowerPoint. Quick change of scenery, though. I did have to go into my basement because we have construction going on in the house. So if you hear any sounds in the background, that is why. And it's raining outside, so I can't really go outside, and they're using saws. So that's that. So let me just get to the PowerPoint. Awesome. All right. So we finished up on talking about incomplete dominance and codominance last PowerPoint. We talked about how those were genetically derived. And now we're going to be moving on to multiple alleles. All right. So, excuse me. All right. So, what is an allele? Just as a recap, an allele is a genetic sequence that codes for a trait, right? And so, what is multiple alleles? It's when a trait is controlled by multiple different coding sections of DNA, right? And so, examples of these are your blood types. Again, blood types are so common in genetic conversations. Coat color in rabbits, whether they're a whole coat or whether they have a body color and then their ears are different colors, for instance, those are different alleles. See for full color. Yep. All right. And so blood types again. All right. And so blood types is controlled by multiple alleles, either your type A, B, AB, or O. And to put it simply, right, if you're type A, you either homozygous allele, so you have two A's, or you're actually AO. Right. You can see that in this chart. Same thing for B, either you're homozygous B, so BB or BO. All right. AB is just heterozygous AB, and then type O is homozygous recessive, right? So reading the third bullet point, it says O is recessive. Two O alleles must be present for the person to have type O blood, right? Type AB blood, A and B are codominant in that situation. If a person receives an A allele from mom and a B allele from dad, their blood type is going to be heterozygous AB or type AB blood. And crosses involving blood type often use I to denote the alleles, right? So you'll see how in this chart under genotype in that left genotype column, they have I's representing those alleles there, right? It is called a superscript when the letter associated with that allele is above it, right? Almost like where an exponent would be on a math digit. All right, so progressing through the PowerPoint. All right, so when doing blood type crosses, you'll need to know whether the A or B person is heterozygous or homozygous, because again, if you're blood type A, you can either be either homozygous A as in AA or heterozygous A as in AO. Right. Type O's are automatically homozygous recessive OO, and type AB is always type AB, right? And so you will see these blood crosses on your screens. So on the left Punnett square, you have a parent blood type AB crossing with another parent type, which is type A, but it's actually heterozygous A, so it's blood type AO, right? And you can see that those blood types for those generations are there. On the right side is just another Punnett square, just showing you how a parent who is type A mates with a parent who's type B, and they're both heterozygous, they can produce a blood type O offspring. All right, so just continuing. All right, and so blood type determines what antibodies are located within the blood, right? And so if you have type A blood, you have type B antibodies, and same vice versa. So if you have type B blood, you have type A antibodies, right? So we know that we all have blood types, and we can only receive the same blood type, right? And that's because of these antibodies reacting with these antigens that stick on the outside of your red blood cells, right? Just reading through the bullet points, all right? Type AB blood has no antibodies at all, and any blood can be donated to them. All right. And that means that they're universal acceptors. So if you're type AB, you actually follow the rule that you can receive any type of blood and be okay. Right. And then type O blood has no surface markers. Those are those antigens. They're like protein, kind of like just like telling out what type of blood it is based off of the protein structures or antigens on those red blood cells. And antibodies in the blood of a type O person, they don't react with any type of blood. So they're considered universal donors. So a type A person, blood type A person can receive type O blood and be fine. All right. So continuing on, right? So this is kind of going into those full coat or it's called Himalayan is the type when they have different colored ears, feet, and a front. And so in the top left, we can see the full color, and below it, each pair of letters is a genotype, all right, with two alleles denoting for each letter. And just in the combination of each allele, you see the different full coat colors versus the Himalayan versus the homozygous recessive trait would actually be an albino bunny. So this is Peter Cottontail on the bottom right. All right. So continuing on. All right. So we have polygenetic traits, right? And so when someone says they're tall, right? Yes, they are tall, but there's a lot of factors that play into what makes someone taller than someone else. For instance, you can have longer legs or a longer torso. You can have just long femurs, but short tibia and fibulas, right? The length of all of your bones that help you stand up vertically is going to affect your overall height. And each part of your body essentially codes differently in your DNA. And so a polygenetic trait is something that is controlled by many things, prefix, poly, right? Polygenetic, many genes, right? So it's when two or more gene pairs control that trait. All right, examples of this are eye color, hair color, skin color. We're going to be talking about skin color in a couple examples soon. All right, we're going to talk about how this is going to show like a bell curve, right? And what that means is that we all have an idea of what a bell looks like, right? And you're going to see how we're going to see data represent a bell shape once we plot it out, all right? And so like this is a bell curve. Right, and so look how this is mapping human height. And so around here we have just about 800 individuals in the middle at 5 foot 5 inches. All right, that's an average height. All right, once you go right or left with that, you'll see how it gets a little bit lower. All right, some people are just 5 feet tall, some people are 6 feet tall. Right, once you go a little bit further away from the middle, you'll see how it gets lower still, right? Some people are 4 foot 5 inches and some people are 6'5", right? We can all kind of imagine someone that fits into all of these categories, 4 foot 5 inches all the way up to 6 foot 5 inches. All right, but look at the two extremes. All right, we don't know many people who are only 4 feet tall, and we don't know too many people who are 7 feet tall, and that is because height is being derived from multiple factors. It's a polygenetic trait. There's a lot of things that affect how tall someone is, right? So we're now looking at skin color as a big pedigree chart, right? So this is multiple different genotypes. Instead of letters, we're actually using these dots that are colored in and such. All right, and so the very top left, you are crossing two people with almost no skin pigment. All right, that's why all three of these for the sperm and egg are not filled in, and you are going to get a very pallid or very pale phenotype when you cross those two people. All right, but the more you go down on the egg side, so on the vertical column, and the further you go to the right on the horizontal column, you get darker and darker skin tones. And so in the very, very bottom right where my cursor is, this is the darkest skin tone possible. All right. And in the middle is all of the mixed combinations of each. Right. And so we can also represent this as a bell curve here. Right. We know that there are African Americans who have very dark skin, and there are very, very Caucasian people who are very, very light skinned. Right. And then the average, the global average, right, is what we're looking at in this bell curve. And, you know, we have so many ethnicities in this world, and we're all made a little bit visually different. And we can see that through skin color on this bell curve. And so most people would be not very like paper white or very asphalt dark, right? White and black. Most people are usually in like the tan range in the middle, right? So moving on. All right, some traits are actually considered our X-linked or sex-linked characteristics, right? Most common, if you have a sex-linked gene, it's going to be carried on the X chromosome. And why is it carried on the X more likely than the Y? Frankly, just the X chromosome is bigger and there's more room to carry certain types of traits, right? And so examples, all right, colorblindness, all right? A lot of people are colorblind and typically they're males, all right? We'll see that's why. Hemophilia is when someone lacks blood clotting factors. So a lot of that has to do with the antigens and antibodies. We talked about those a little bit earlier. And both are recessive and both are carried on the X chromosome, all right? And so in this plot, all right, in the top left, you should be able to see a 5, all right? I dragged my cursor over it, all right? In the top right, you will see a 7, all right? And in the bottom, you should see 42, all right? And if you didn't, get your eyes checked out, all right? Colorblindness, all right? So determine the genotypes of the following. This is supposed to be an in-class, break out your pen and paper, but we're just going to go through this, okay? All right, so a man that is colorblind, all right? Again, it's carried on that X chromosome, so this X little c Y, all right? That is going to be a colorblind man, all right? How about a woman who is colorblind? What do you think those are going to have? We know that a woman sexually is coding for XX, right? Males code for XY, right? So a woman who is colorblind is actually going to have 2 X's with little C's on them, all right? And then I think we finished with a carrier. So a woman who's a carrier will have X big C, X little c, and we see that right here, right? Again, you're looking at those superscripts, right? And this is just a cross, all right? So a father with hemophilia will cross with a mother without hemophilia. And because the mother is passing down normal X chromosomes, no one's going to have hemophilia, but 2 of the daughters will be carriers, and the males are actually going to be fine because the father passed the Y chromosome to the males, the sons, and they don't have that X chromosome with that mutated gene on it, all right? And so this last slide is just the mass state standards for what we need to know for genetics, all right? And so just going through them very quickly, we have to develop and use a model to show how DNA in the form of chromosomes is passed from parents to offspring through the process of meiosis and fertilization in sexual reproduction. We did that, all right? We're good with meiosis. We've talked about phenotypes and genotypes and heredity. All right. Second thing, make and defend a claim based on evidence that genetic variation or alleles may result from new genetic combinations via processes of crossing over and random segregation of chromosomes during meiosis. B, mutations that occur during replication, and or C, mutations caused by environmental factors. All right. We did touch upon mutagens a little bit, all right? If anything, I could give you a little bit more on that, but as long as we know that because of random assortment in meiosis, we have genetic variation because it's random, all right? Third one, apply concepts of probability to represent possible genotype and phenotype combinations in offspring caused by different types of Mendelian inheritance patterns, right? So we know how to do Punnett squares. We've discussed pedigrees and diagrams and such. So I think we're pretty much good there in figuring out and drawing out the probability of heredity, or just making a Punnett square essentially. So hopefully you can do that. And this last one, number 4, use scientific information to illustrate that many traits of individuals and the presence of specific alleles in a population are due to interactions of genetic factors and environmental factors, all right? And so we honestly haven't talked about how the environment can affect your genes. Maybe I'll go into that a little bit in a next video, okay? All right, but for today, all right, that is the end of the PowerPoint, and I will be posting this PowerPoint alongside this, all right? And so let me just get back to my video. All right, I'm going to stop presenting. All right, almost. All right, and so that is the 4th genetics lecture PowerPoint. I'm sorry it was a longer video, but I really did want to make sure that we touched upon all of the important parts before you guys do that Albert I.O. So with this video, I'm posting an Albert I.O. assignment, which is quote unquote graded. All right, this is an assignment posted past April 6th. And so I can give this an actual grade. All right. So please do the Albert IO. Again, answer all of the questions, whether you completely bomb the assignment and get like a 30% or lower. As long as you complete all the questions, you will get a submitted grade for it. All right. So please watch the videos, take some quick notes, internalize some information, think about what it is to have genetic information inside of you, and then complete the Albert to the best of your ability. All right. Please email me if you have any questions. All right. Thanks so much for your time, everybody. Do your homework. All right. Thanks, guys. Bye.